Today I'm going to talk about the urinary system. I'm going to give a brief overview after you've already seen this in lab. Talk about the workings of the nephron, which is the functional unit of the kidneys, and the processes that they go through for the formation of urine. In this video, I'm going to focus on the first part of that, which is what's called glomerular filtration. Now, the urinary system, I know you're familiar with from lab already. If we could remove most of the abdominal viscera, what we'd see is this kind of appearance, where oftentimes one kidney will be higher than the other one. They receive their blood flow directly from the aorta, from their own renal arteries, and then the urine that's produced at the end of this process that we're going to talk about today is going to get sent down the ureter to get stored in the bladder until voiding happens. Now, because you've done the dissection already, you know that this picture doesn't look exactly like the cat did, because you recall that the kidneys have this kind of retroperitoneal placement to them. So if you are looking from the underside here, because we're looking at the bottom of the diaphragm up here, you can see basically there's a muscle. There's not just behind the muscle wall, but there's the layer of the peritoneum that's going to be separating it from the rest of the abdominal cavity. I think we've talked about that a lot in class and lab, so I'm not going to belabor it anymore here. But just as a reminder of how high they are, we tend to think them, at least I tend to think about them as being pretty low down. But remember that those last two floating ribs are what's going to be those last two floating ribs are going to be covering over the kidneys. Now, when you look at the kidney itself, again, just by way of general review, we're going to divide up the kidney into cortical regions where most of the filtration happens, and then the medullary area, which is divided into a series of pyramids and with columns in between each of the pyramids that are dividing those up, and that's where the reabsorption and secretion is going to happen in the process of forming the kidney, forming the urine. Hey everybody, today we're going to start to talk about the urinary system. I'm going to give a little bit of an overview of the kidneys and then start to talk about like the overall structure. Hey everybody, I'm going to talk about the urinary system today. A little bit of an overview of the kidneys in particular, talk about the structure of nephrons, the different kinds of nephrons, and the functions that they have in forming urine. Now, the production of urine is kind of a complicated process, so I'm going to divide that up into a couple of parts. The first part of it is what we call glomerular filtration, which is what I'll also cover in this video. The next video will include stuff about what happens after the filtrate has been formed, but before it turns into the final urine. So you've seen the urinary system before, so I don't need to belabor this too much. Recall that the kidneys are like relatively high up in the abdominal cavity, but they have this retroperitoneal placement, meaning if we were to look now we're looking from the bottom up toward, like, say, where the diaphragm is up here. You remember when you dissected the cat, you had to sort of pick through the back of the peritoneum in order to get to where the kidneys themselves were. So, and also this also reminds me that, because they're up pretty high, they're actually covered by most of those last two floating ribs. Now, you recall also the kidney, we divide up into corticular regions out here where the filtration happens, and then the medulla where the filtration and reabsorption is taking place. Most of the parts of the nephrons that are in there are within these renal pyramids, but the pyramids, the spaces in between them where blood vessels pass through, are called the renal columns that are in between. And then both the renal artery and vein, as well as the ureter, all come off of that kind of in-out area that we call the hilum of the kidney. If we were to look specifically at the blood flow pattern, this is actually a pretty helpful figure for reminding us of how things are divided up. So first, the renal artery brings blood in, and then that renal artery splits into a bunch of smaller vessels. Those first vessels that come up are what we call the segmental arteries. The segmental arteries continue to split, and then at the level when they pass through the renal columns, they're referred to as the interloper arteries. I've seen interlobular before, but we'll use interloper arteries. The interloper artery then splits and goes along like what would be the base of the renal pyramids and goes into this kind of a arch-like structure that we call the arcuate artery. You recall when we were looking under the microscope, we used the arcuate artery as the border between the cortex and the medulla. And then finally, what sort of goes out directly toward, like perpendicular to the surface of the kidney, those are what we call the cortical radiate arteries. Now, those cortical radiate arteries, what's sort of boxed in here, is the blood vessels that are kind of go right around the nephron. We'll deal with those when we get there, but suffice it to say that the cortical radiate artery branches into what's called the afferent arteriole that leads to this fenestrated capillary called the glomerulus, and then the efferent arteriole leaves the glomerulus and then turns into the vessels called either the peritubular capillaries or the vasa recta, depending on where we are exactly. And those will go to one place or another. Either to the cortical radiate vein or to the arcuate vein, and then kind of work their way back through the columns of the interlobar veins, then eventually to the renal vein to the inferior vena cava. Now, the renal corpuscle itself has a couple of parts. First, there's this little tuft of capillaries that's called the glomerulus. Now, this is formed of fenestrated endothelium, so they're very porous, so they allow for efficient formation of filtrate. Now, the filtrate, what you probably remember from the different kinds of capillaries is that fenestrated capillaries are the ones that have small pores that allow substances, solutes in particular, that are a lot smaller than a cell to be able to get filtered. And again, filtration is the process where hydrostatic pressure squeezes fluid out from the plasma onto the other side of the capillary. That plasma that gets squeezed out into the space on the outside of the glomerulus, we're going to call filtrate. It's not urine yet, but it's like this rough draft of the urine that's going to get modified later on. Around the renal corpuscle, the outside of the renal corpuscle is a structure that's called either the glomerular capsule, or older literature calls it Bowman's capsule. It's sort of a cup-shaped hollow structure that surrounds this little capillary. And it's got a couple of layers to it, much like we'd see if we were looking at a little visceral membrane. So there is an outer parietal layer and then an inner visceral layer, again, just like what we'd see in a serous membrane. But what sits right on top of the glomerulus itself is a series of cells that are called podocytes. Now, these podocytes are little branching epithelial cells where these extensions sort of end in these structures that foot processes that cling to the basement membrane. And then the filtration slits between those foot processes allow the filtrate to pass into the capsular space. If we zoomed in really closely here, you'll see like these really kind of filigree-like foot processes as opposed to the main body here. But then what passes in between, so if over here is the glomerulus, and on this side is the basically the space in the capsule, you see there are like these little diaphragms that almost get set up where the filtration slits don't exactly all line up with where the foot processes are, but it allows substances to get squeezed through and then also not leak back again. Now, after the glomerulus is the renal tubule and the collecting duct. The renal tubule is about 3 centimeters long and consists of a single layer of epithelial cells throughout. However, each region has its own unique histology and function. There are a few main parts that we learned about in lab. First, the proximal convoluted tubule, called proximal because it's close to the renal corpuscle, the Nephron's loop, and then the distal convoluted tubule. So both the proximal and distal convoluted tubules are situated in the renal cortex, but the loop and part of the collecting duct, which is a shared structure by a bunch of nephrons, appear in the renal pyramids, which is what gives them that sort of grain-like shape. If we look at the histology of the different parts of the tubule, the proximal convoluted tubule has cuboidal cells with dense microvilli that form a brush border, like the brush border that we saw in the duodenum. And also, as in the duodenum, that helps to increase the surface area. They've also got a whole bunch of mitochondria because they are relatively metabolically active cells that are both absorbing and secreting substances that are in the filtrate. The nephron's loop has a couple of different parts. There's a descending limb, and then there's like this thin part where it loops up again to the ascending limb. These can be cuboidal or columnar cells as you get into the distal part, but they tend to be more squamous when you're looking in the thin segment of the loop. The distal convoluted tubule has cuboidal cells with very few microvilli. In this case, they function more in secretion than in reabsorption, so they don't need the villi as much. And again, this is kept up here in the cortex. Then after this comes this shared structure, the collecting duct. There are a couple of principal cell types. One we call principal cells, which are short, that have, they're every now and then with short microvilli, and those help to maintain water and sodium balance. The rest of them are intercalated cells, which are cuboidal cells that have lots of microvilli, and they help to maintain the acid-base balance of the blood. So the collecting ducts receive filtrate from many nephrons, and they are going to fuse together ultimately when they get down to that renal papilla that's going to release the urine into a minor calyx. There are two different kinds of nephrons. One is called a cortical nephron. And if you look at the figure here, you see there are a couple of differences where the cortical nephrons, as the name suggests, they're more up in the cortex. These make up like 85% of the nephrons in the whole kidney. They have a long proximal convoluted and distal convoluted tubules and relatively short loops. And notice how most of the capillaries that go around them, most of what we call the peritubular capillaries, are going to be situated around the proximal and distal convoluted tubules, but not as much around the loop. On the other hand, the juxtamedullary nephrons, sort of like the word juxtapose means like it's close to. Juxtamedullary nephrons, they have a really long loop of Henle, and really long nephrons loop, and and also the peritubular capillaries around it are what we call the vasa recta, which helps to form the process of concentrated urine. So most of the concentrated urine comes from the comes from these juxtamedullary nephrons rather than the cortical ones. Now, just to go back to, if we could divide up the functions that happen in the nephron, I like this cartoon here for of the world's simplest nephron. So a given cortical radiate artery gives rise to the afferent arteriole, which then goes around the glomerulus. And then the efferent arteriole is going to go to some kind of peritubular capillary. Again, either the ones that are around the PCT and DCT or the vasa recta that's going around the nephron's loop. Either way, those are going to be receiving some substances through tubular reabsorption, or some substances are going to get dumped back into the filtrate, and eventually it's going to turn into urine by the time it's at the end of the tubule system. Then this blood is going to continue on to the cortical radiate vein and eventually ultimately to the renal vein. But to focus in on now the process of urine formation at the beginning, around the renal corpuscle there are a few different populations of cells in addition to these podocytes that we already talked about. So I'm going to talk about the structure that altogether is called the juxtaglomerular apparatus. The juxtaglomerular apparatus includes three different cell populations. The first one is the cells that are called the macula densa. So these are these tall, closely packed cells of the ascending limb of the loop, and they contain chemoreceptors that sense the salt content of the filtrate. So these are chemoreceptors now that are sniffing for sodium ions. The next are cells that are called granular cells. Now these granular cells, they are enlarged smooth muscle cells of the afferent arteriole, and they act like mechanoreceptors that sense the blood pressure in the afferent arteriole. So particularly, If the blood pressure is too low in that capillary, it releases the hormone called renin. So the renin-angiotensin system, the renin that we've been talking about all along coming from the kidney, comes from these cells. So when blood pressure is too low, renin is released by these cells, which eventually is going to cause systemic blood pressure to go up. Then, last of all, in this sort of triangular-like shape here, is what's called the extraglomerular mesangial cells. So these are located, they're interconnected with gap junctions, and they seem to pass signals between the macula densa and the granular cells. So you've got both mechanoreceptors here, chemoreceptors over here, but those chemoreceptors have a way of communicating with the mechanoreceptors in the granular cells. Now, the critical thing about producing urine, like we process tons of urine over the course of, tons of blood over the course of the day. The book claims that 180 liters of fluid is processed daily. However, only about, like 1/100th of that, only about 1.5 liters or so of urine is produced any given day. So the kidneys filter the body's entire plasma volume at least 60 times a day and consume about 1/4 to 1/5 of the oxygen of the body at rest. Now, the filtrate that's initially formed up here in the renal corpuscle is basically blood plasma minus some proteins. Now, the urine is what, as I said before, is what's produced from the filtrate, but only about 1% of the original filtrate really turns into urine, because the stuff that's in the urine is going to be concentrated in like metabolic wastes and any other substances that are not needed anymore. So, just to kind of walk through the process a little bit, glomerular filtration is an entirely passive process where hydrostatic pressure forces fluids and solutes through the filtration membrane into the glomerular capsule. But there's no reabsorption into the capillaries of the glomerulus because of those podocytes that sit on top of them. Now, when you look at this figure, this is kind of modified from the original figure when we were talking about hydrostatic pressure and and moving the fluids out of capillaries. So, as before, there are a few different pressures that affect filtration. First of all, they're the ones that are pushing the fluid out. And before, as just like we talked about before, it's hydrostatic pressure that's in the glomerular capillaries is what's pushing the chief force pushing water and other solutes out of the blood. It's actually quite high compared to other capillaries in the body. It's around 55 millimeters of mercury. So it's about twice as high as what we see in most other capillary beds. The reason for this is the efferent arteriole is like a high resistance vessel with a diameter way smaller than the afferent arteriole. So as a result, there's a higher hydrostatic pressure pushing out there. But as before, there are some substances that are going to be sucking the fluid back in. There's the colloid osmotic pressure. That's what the sucking force is of the proteins in the blood. And there's also this fluid of the outside, the pressure of the hydrostatic pressure of the fluid within the capsule, which is going to be relatively high, but not as high as the glomerular hydrostatic pressure. So like the sum of all these is what we call the net filtration pressure, the NFP. So it's basically it's fair to say that the hydrostatic pressure in the glomerulus, that is the main factor that determines the glomerular filtration rate. If GFR is really high, then there are problems, and there are also problems if it's too low. So just to kind of walk you through this, if the this is a big figure that's in your textbook that you're going to kind of want to work through on your own, but suffice it to say, the glomerular filtration rate is the volume of filtrate that's formed per minute by both kidneys. Normally, it's going to be around 122 milliliters per minute or so. The glomerular filtration rate is directly proportional to the net filtration pressure, which is primarily the hydrostatic pressure coming out of the glomerulus, the total surface area available for filtration. So you figure that through your life, your kidney function will decrease bit by bit. But also it's the glomerular mesangial cells that help to control that total surface area by contracting. And then last, the permeability of the filtration membrane. So and again, the glomerulus is much more permeable than most other capillaries we see. Now, it's important for us to be able to maintain a constant GFR glomerular filtration rate, because it allows the kidneys to make filtrate and to maintain their extracellular homeostasis. The goal of local controls, what we call the intrinsic controls listed here, these help to maintain the GFR in the kidney by regulating the systemic blood pressure. So, for example, if systemic blood pressure goes down, that's also going to decrease the pressure of the afferent arterioles. So you decrease the stretch of the smooth muscle in those vessel walls, which will cause them to dilate. By dilating those, that's going to help to send more fluid through the glomerulus, so that's going to increase the glomerular filtration rate. Again, so that's called the myogenic mechanism. In addition to that, there's also the tubular glomerular mechanism of autoregulation, which is, as GFR goes down, there's a decreased flow of filtrate through the loop. So those macula densa cells that are sensitive to sodium, they're going to start to communicate through those central cells and cause the vasodilation to happen in a similar mechanism. By opening those afferent arterioles, it's also going to make GFR go up. Okay, so we got two different routes so far that are intrinsic to the kidney, right? So if you decrease blood pressure, the blood vessel will dilate because of the muscle stretch, or if you're producing less filtrate, less sodium going through, the macula densa cells will also communicate to the afferent arteriole to make them dilate, both of which are going to increase GFR. In addition to that process, there are also things that happen through hormonal mechanisms. I mean, primarily this is going to be the renin-angiotensin system. So, as you saw before, those granular cells, when blood pressure goes down, granular cells of the juxtaglomerular apparatus are going to release renin. Renin is going to catalyze this reaction eventually so that angiotensinogen turns into angiotensin II, which is going to make a couple things happen. Angiotensin II acts on systemic arterioles, causes vasoconstriction, which is going to increase the total systemic blood pressure. Now, this is different from the vasodilation that's happening over here. This vasodilation is happening only of the afferent arteriole. So if you increase the bore size here, you're going to be able to push more fluid through. On the other hand, down here, this is causing vasoconstriction elsewhere in the body. That's going to help to increase the systemic blood pressure overall. Okay, so that's one route. The other route is angiotensin II, we know, is going to stimulate the release of aldosterone, and aldosterone is going to actually go to the kidney tubules. It's going to cause reabsorption of salt. Now, here's the thing about sodium. One of the rules I think we mentioned before is that water follows sodium. So by reabsorbing sodium, that helps to suck in more water again from the filtrate back into the plasma. So as a result, it's going to increase your blood volume. So that is also going to increase your systemic blood pressure. In addition to these, so we have these direct mechanisms that happen inside the kidney. There are extrinsic mechanisms that take place elsewhere in the body, but have the result of propping up systemic blood pressure. Then last here are these neural controls, where baroreceptors, say in the carotid sinus, are going to inform the brain of lower blood pressure, which is going to turn on the sympathetic division of the ANS. And it's going to cause the vasoconstriction of these systemic arterioles, which again is going to prop up blood pressure. Okay, so the big thing about this figure is that the kidney has to its avail a bunch of different ways to let glomerular filtration rate be stable. Some of these are caused by Many of these are caused by cells that are right around it in that juxtaglomerular apparatus, but others are going to be controlled by the brain as well. So that's the main thing for this first part, is talk about filtration. Now we're going to get into tubular reabsorption and secretion, which is the part that happens later.